Students know. Good morning to you all. Today our class is of transport of things across the cell membrane. As in the last class, what we have seen, we have all read about the plasma membrane, isn't it, which surrounds the cell. So this cell, it is a bilipid layer. In between, you have got some protein molecules from where the things can pass from one extracellular fluid to the intracellular fluid. This is in the last class we have read. They are hydrophobic and hydrophilic. Now, as you see, the gases such as oxygen, carbon dioxide, and nitrogen and alcohol, they can pass through the cell membrane very easily. Without any disturbance, they can flow inside. Other molecules, hydrophilic molecules, benzene, they can also flow from one side to the other side very easily. And smaller molecules, these smaller molecules, that is water and ethanol, they are small polar molecules. They can also pass freely from one end to the other end. When you come to the larger molecules, that is glucose, because it's having a molecular weight is more and the size of the molecule is also big, so it cannot pass easily from the cell membrane. Isn't it? So it has to have a gate or a through gate, it can easily, gated channels through which it can easily pass. Same thing as charged molecules, that is amino acids, sodium chlorides, ions. All these things, they also cannot pass through very easily. So, plasma, it retains dissolved material, allows exchange of material. Dissolved material will be inside only, and it also even allows the exchange of materials. Two types of movement are there. One is passive. Passive doesn't need more of the energy, so it can pass from one side to the other side very easily. Examples such as from an upstream river, the flow of the water can go down very easily, passively, without needing any energy. But if you want an active side, that is, energy has to go against the gravity from down to up. So it needs energy. Such as even in the houses you can see we have got for bore there are pumps so that water from the below has to come up, but water from above to the down it can come very easily. That is called as passive transport, and this is called as energy. Active transport. Active needs energy. Passive doesn't need energy. Both lead to the net flux of an ion or a compound. So, by both the means, there can be a net influx or efflux. So, classification. Now comes to the passive transport. Simple diffusion through bilipid layer, that is through the proteins channel also. Diffusion in between the bilipid layer, you will find the protein molecules. These molecules are just like gates. They allow certain things to pass through it and certain things they cannot pass through it. So it is a gated channel. Second is now diffusion through the protein channel. Then osmosis. Osmosis, I think you have all read from the 10th class itself. That is a concentrated water and a solute and a solvent. In between there will be a semi-permeable membrane through which solids they pass towards the liquids. That is ions and other things they pass towards the clear solution. So that there should be an equilibrium. And other method is facilitated diffusion. Facilitated diffusion means it needs some facility, some help it needs so that it can diffuse from one side to the other side. Now, second thing is that again it's an active transport. As I told you, it needs pumps, sodium potassium pump, sodium calcium pump, and so all these pumps are needed for the transport of in the active transport. That is pumps which are primary and secondary. Then endocytosis. Endocytosis means within the cell, as we have seen, we have got ribosomes. Ribosomes, what they do? They synthesize protein. These proteins, once synthesized, where they will go from the sarcoplasmic membrane into the Golgi apparatus. So, as you have seen, proteins which are collected in the sarcoplasmic produced they go into the vesicles. From vesicles, they go to the storage place, that is Golgi apparatus. So, whenever required, it comes out and again forms a vesicle and it goes towards the cell membrane and it passes out where it needs an energy. So this is called as endocytosis, means from outside to inside. From inside to outside is exocytosis. That is what we have discussed, what we were discussing is exocytosis. From the cell inside, from the Golgi bodies, if it has to go out, it has to go towards the cell membrane, proteins and other things. Then they have to pass from inside the cell, that is intracellular, to extracellular fluid. Same as endocytosis. From extracellular fluid, they have to come into the cell. In that way, there is endocytosis and exocytosis. One has to go out from the cell. Exo, one has to come inside the cell. Endocytosis. Movement of substances across the cell membrane. Analytics of the solute movement. Uncharged particles move due to the concentration gradient. Wherever the concentration is less, from higher concentration towards the lower concentration, they move, or uncharged particles. Charged particles, two gradients. One is it will go through the chemical gradient. Second is through electrical potential gradient. And sometimes it goes in a way that is to form a electrochemical gradient. So in this way, the things can pass from the cell membrane. So energetics of the cell as it is, that is energetics are there. So it will move from the higher region to the lower region, it will move by its electrical movement. And wherever there is a lesser concentration from higher concentration towards the lesser concentration also, they move with their own energetic movement. They are both charged particles and uncharged particles. Simple diffusion. What is simple diffusion? Diffusion is the net movement of a solute molecules or ions from the region of their higher concentration to their lower concentration. So if you have taken a that is container, in between the container, if you put a semi-permeable membrane, at one side, if you put a solute and other side, you put a solvent. In the solute, if you have got glucose molecules, these glucose molecules, they form a higher concentration. So other side it is only a solute. So from higher concentration, these glucose they will pass on to the other side, so that there should be an equilibrium. Molecules move from the concentration gradient. Molecules have kinetic energy which make them move about randomly. As a result of diffusion, molecules reach an equilibrium where they are evenly spread out. Once it becomes an evenly spread out solvent and a solution, when they become equilibrium stage, at that time there will not be any movement. Simple diffusion, as you have seen, from the outside, if the cell membrane is very much having glucose and other molecules, which they have come from the lumen of the intestine and they have come towards the extracellular fluid. From the extracellular fluid, they go and to the intracellular fluid. Isn't it? From extracellular fluid, they are going to the intracellular fluid, then into the bloodstream. From here, that is going to the bloodstream. The molecules move down the kinetic, which makes them move about randomly. So, here is an example. Blue particles, which are mixed, they are glucose molecules, and now they are moving here from one membrane to the other membrane. That is inside cell, outside cell it is only a solution, here it is a solvent inside. So, diffusion starts, simple diffusion starts from the semi-permeable membrane. Inside the cell it is concentrated, outside the cell it is not concentrated. So there is a movement diffusion that is going through the semi-permeable membrane from inside the cell to the outside cell. Once the equilibrium is reached, both the solutions, when they reach the equilibrium, at that time there won't be any exchange of the material will be there. Determinants for the rate of diffusion. So in which conditions the rate of diffusion will be more, and in which conditions the rate of diffusion will be less. So these are the things which you need to understand. Steepness of the concentration gradient. The bigger the difference between the two sides of the membrane, the quicker the rate of diffusion. Say if you are having a small room and a big room, that is from upstairs to downstairs, it can easily, concentration gradient can easily move. We are talking as an example. So from higher side to the lower side, it can be very fast. Temperature. Higher temperature gives molecules or ions more kinetic energy. Molecules move around faster, so the diffusion is faster. For which I can give you an example. That is, whenever the temperature rises. Whenever the temperature rises means when you do exercise, at that time there will be temperature rises in the body. So whenever in the fever also temperature rises. So whenever you do exercise, at that time there will be temperature increase. As the temperature increases, movement of the molecules also increase more. They move around faster, so the diffusion is faster. So anything in the cold region will have temperature is less, and their movement will be less. As you see, usually we will be lazy in the winter season, whereas we are not lazy in the summer season. We get up early, we do some work and other things. As such, the temperature, even on the other side, if you're not only from the molecules side, even if you see as an example in our own life, at higher temperatures we do more work. In lower temperatures we never try to come out from the house and do any work because it's too cold. Surface area. What is this surface area? If you are having a sari, the surface area of the sari is bigger. But when you pleat it, when you make it into pleat, the surface area becomes smaller. So if the surface area, the greater the surface area, the faster the diffusion can take place. This is because more molecules or ions can cross the membrane at any moment. If you are restricting the movement, that is, if you are restricting the area, the movement will be slow. If you are extending the area, that is, you are opening the pleats of a sari, your surface area is getting increased. In the same way, the surface area where there is a bigger surface area, the movement of the things will be more faster. The surface area is small, the movement will be slow. The types of molecules are ions. Larger molecules need more energy to get them to move, so they tend to diffuse more slowly. The larger the molecule, it will be slow will be the rate of diffusion. The molecules are smaller, diffuse more slowly. The non-polar molecules diffuse more easily than polar molecules because they are soluble in non-polar phospholipids. That are, as you have seen in the beginning itself, non-polar things, that is oxygen, nitrogen and carbon dioxide, they can pass through the phospholipids very easily. They are polar molecules because they are soluble in the non-polar phospholipids. They can pass very easily, whereas polar molecules, sodium, potassium, they are having bigger molecules, even glucose, amino acids, urea, all these things, they are bigger molecules. As you have seen, albumin, the molecular weight of an albumin is 69,000 molecular weight. So in that way, the molecular size is bigger. If the molecular size is bigger, the rate of diffusion is slow. If the molecular size weight is less, the diffusion is fast. Molecules that diffuse through the cell membrane. Lipid soluble molecules. What are they? Oxygen, carbon dioxide, and water. They can diffuse very easily because they are lipid solubles. That is from outside to inside or inside to outside. They can carbon dioxide, but oxygen, non-polar, so diffuses very quickly. Carbon dioxide, polar but very small, so diffuses very quickly. Water, it's also polar but also very small, so diffuses quickly. Diffusions of ion through the plasma membrane. That is a bilipid layer highly impermeable to sodium, potassium, calcium, and chlorides. But their conductance is critical. But it has to, one way or the other, it has to pass. So ion channels provide mechanism for their transport. What are the ion channels? These channels are found on the bilipid layer. Divisions of ions, the most channels exist in an open or closed conformation. The term it is called as gated channels. So gates, as the gates open, they can enter in. The gates are not open, they cannot enter. So they are called as gated. Now we are seeing gated communities. In the same way, even our body has got, that is bilipid layer has got gated places from where it can, all these ions can enter in. They are open to some ions. They are open to, some are open to other ions, that is sodium, potassium, chlorides, all these things, they are different types of gates are there. Some, whenever there is a need, the voltage gated channel, during the action potential, there is a change in the voltage. So voltage gated, at that change you can see that is gates open and enters in. For that example is whenever there is a stimulus, action potential changes. So what happens? Chemicals from the ions from the this one axon terminal, that is from the nerve terminal, that is from axon, it is coming down towards the lipid layer from where it is these ions they come out, that is into the channels. So in these channels, when the gated channels open, they can enter in because acetylcholine, the these vesicles have got acetylcholine. These acetylcholine they come and they are on the receptor sites are out there for the on the gated channels. The receptor sites are there. So these receptors they receive the acetylcholine. Once the acetylcholine is received, they open the channels. So it goes inside and there is a movement of sodium and potassium. Isn't it? So in that way, gated channels, they are their voltage gated channels and ligand gated channels. Ligand gated channel means hormones and neurotransmitters or any enzymes. If they are there, they can open the channel, such as insulin. Insulin that is just below the bilipid layer, there are vesicles that receptor sites. These receptor sites they consist of insulin. They easily, when insulin is there, sugars can come down easily. If insulin is not there, it cannot, sugars cannot be come easily come down into the cell. From there it has to go out and go into the blood. So for that ligand gated channel they need hormones. Hormones such as insulin, and for here the voltage gated channel they need acetylcholine, so that any voltage change of voltage the gates will open and the receptor sites they receive the acetylcholine and then the movements go on. Is it clearly understood or still if you are having any doubt, when you come to the action potentials, at that time you can learn more. This is only just in the beginning we are explaining how many types of gated channels. There are voltage gated channels and ligand gated channels. These are ligand gated channels. As I have told you, they are need for hormones and neurotransmitters. And voltage gated channels, whenever there is a change in the voltage, that is stimulation is there at that time, then those changes will take place, so that ions from inside they go out, from outside they come in. That such as sodium ions they go outside, and potassium ions they come inside. Diffusion of ions through the membrane, voltage gated channels, potassium channels. As just now I was telling you, whenever there is any stimulation, the potassium has to go inside and sodium has to come outside. Three potassium molecules they go inside and two sodium molecules they come out. So as you see, the potassium they are going down. Once the potassium is going down, sodium is coming out. So in this way, water is coming out and potassium is going in. That is, along with the water it is going inside, but water is again coming outside. Potassium is going inside. How it is going? It is making a way. Again, once it is inside, this is outside, the gate will again close. The bulk flow of water from an area of higher concentration of water molecules, high water potential, to an area of low concentration of water, low water potential, across the partially permeable membrane, that is semi-permeable membrane. Water moves much more rapidly through the membrane than solutes, that is aquaporins, endosmosis, exosmosis, and osmotic pressure. So, when aquaporins are there, water is moving in and out very easily. As I have told you, endocytosis, that is coming inside and going outside, exosmosis. So, when you want to see that this doesn't happen, you have to apply on the osmotic pressure when it is moving easily. If you want to stop it, you have to put a pressure on that, so that there should not be any movement. That pressure is called as osmotic pressure. So, if things are moving, so you put a great pressure on it. That is, you apply a pressure so that there cannot be any movement. That type of pressure is called as osmotic pressure. Once there is no movement, that is called as osmotic. Dilute solution and a concentrated solution. Sugar molecule, that is glucose molecule, once it is coming from outside, that is inside the cell, here concentrated solution, that is outside, here it is dilute solution. So there is a diffusion is going on till the equilibrium is met. If you want osmotic pressure, you have to put a pressure on the molecule so that it should not move. So in that way, if there is an osmotic pressure, there won't be any movement. Osmosis, as you are seeing, this is the from higher concentration to the lower concentration. Equilibrium, once it is equilibrium is maintained, there won't be any movement at all. Or the net movement is already over. As you see, if you are putting cells placed in a distilled water, they will swell. If you are putting them in a salt solution, at that time, the water from inside will come outside, because outside it is more concentrated, inside it is less concentrated. So it will shrink as you have seen from higher concentration to a lower concentration. So this example, is it easy or not? That is, cells which are placed in the distilled water, what will happen? The cell is having more of the concentration. So water is going inside the cell and it is getting swollen. And after getting swollen, if still it imbibes more water, at that time it bursts. Here is the opposite thing. The cell is shrinking because the outer solution, that is salt solution, is more concentrated than the inner solution of the cells. So it is taking out the water from the cells. So it is shrivel. Cells shrink and shrivel. Facilitated diffusion. How is the facilitated? So it needs some facility or some help it needs so that the diffusion can take place. Substrates selectively bind to a membrane protein. As I have told you, there are membrane proteins for gated channels for sodium, potassium, calcium, and other things. When you come towards uh, muscle now, at that time you see how there is a sarcoplasmic tubular system. So this sarcoplasmic tubular system, at the end of it, there are systems. These systems are connected to the cell membrane up, that is bilipid layer. There they have got a gate. So whenever any diffusion enters that sarcoplasmic reticulum, they enter through the sarcoplasm, that is the longitudinal tubule. So once into the longitudinal tubule, they go towards the transverse tubules, as we have seen, microfilaments and microtubules inside the cell. In the last class, we have seen there are inside the cell, there are microfilaments and microtubules. Through these tubules, these can easily come inside and they are helping the transport. In the last class, within the cell, you have seen Golgi apparatus, that is, then you have seen about all these things, that is microfilaments and microtubules. So facilitated diffusion. Here you are seeing from outside how they are coming inside the gated channels. Glucose transporter. It needs a transporter system. From glucose outside, it is going inside the cell. Once it is going inside the cell, again it is closing. Again it opens for the glucose. That is glucose transporter system. So glucose is transported from outside to inside through the channel, and there is a need of transporter. As I have told you, we need insulin. Facilitated diffusion. If it is there, it can easily move from one side to the other side. If it is not there, so the facility will be less. That's why any hormone, wherever the hormones are needed, at that time there can be easily exchanged, such as in the pancreatic enzyme, amylase, and all these things, that is the salivary gland, all these things they can move very easily. As I have told you, the glucose is entering this is interstitium. Once it is coming into the cell from outside, it is coming because here are the receptor sites, that is the interstitial cell. From here, it is coming into the blood, is coming into blood glucose, from where it is entering the blood. From the blood, it is going to the different parts of the body and getting stored at liver, isn't it? So there is facilitated diffusion. How it takes glucose from outside, that is from the gut, it comes onto the interstitial cells, extracellular fluid. From the extracellular fluid, it goes into the intracellular fluid. From the intracellular fluid, it is going into the blood. So, blood low in glucose. So, once it is entering in, glucose becomes high in the blood. So, blood glucose level increases and it is going into the blood and getting stored in the places. So, this is called as facilitated diffusion. Is it clear to you now? From everywhere, wherever you are coming, that is from the intestine also, anything that is coming inside due to the help of enzymes, it is coming to the extracellular fluid. From the extracellular fluid, it is going intracellular, and from intracellular, it is going into the blood. These are carrier proteins. They do not extend through the membrane. They bond and drag the molecules through the bilipid layer, release them onto the opposite side. These carrier proteins, they only carry the proteins from outside to inside. There are no channels as such. They do this. They do not extend through the membrane. They bond and drag the molecule through the bilipid layer and release them on the opposite side, as you are seeing in the diagram. Now, this is an active transport. Active transport needs energy. Any energy, how you will get it? This energy is hydrolysis of ATP, adenosine triphosphate. So, once the phosphate is released, it becomes adenosine diphosphate. This released phosphate is acting as an energy. So, this you see mostly in the contraction of the muscles. Whenever there is a muscle, they contract. How they contract? They have got a active transport. They bring in from the outside, well, potassium is coming inside and inside potassium, the sodium is going outside. So in this way, they need energy. This energy is brought from the breakdown of ATP, that is adenosine triphosphate to adenosine diphosphate. And once phosphate bond is released, again it becomes, after the energy is released, there is a movement. This energy is for the movement sake of these molecules. After that, one more adenosine triphosphate is added to it and it becomes adenosine diphosphate becomes triphosphate. Primary active transport. This is primary active transport. The carrier molecule, it brings it inside. That is called as symport. Once it is going inside, transported from outside to the glucose is transported from outside to inside. Antipode. Once it is coming from outside to inside, one molecule from inside is also going outside. That is called as antipode. Sometimes single thing will come down, sometimes two things, that is sympode. Antipode and sympode, you know now. Antipode is one ion is coming inside and one ion is going outside. So, how it is? This is through the energy. So, primary active transport and secondary, as you have all learned in previous discussion also, sodium potassium ATPase, termed sodium potassium pump. So, drives the transport of ions from each ATP hydrolyzed. 3 sodium ions are pumped out and 2 potassium ions pumped in. Hydrogen ion goes outside the cell. Potassium ion is bringing inside the cell. This is by the help of potassium pump, sodium potassium pump, where for the energy to get you have to release, there should be a hydrolysis of the phosphate bonds. Active transport, ATP is released inside the, that is ATP is broken down to ADP. Again ATP, how it is formed, one molecule of phosphate is again added to diphosphate, it becomes triphosphate. Adenosine triphosphate, it gets broken down to adenosine triphosphate. One phosphate bond is released. So this pump, so at that time there is a opening of the gated channels. These gated channels open. At that time you see two sodium channels, sodium ion, three potassium ions from outside to inside, and two sodium ions, three sodium ions going outside. Now active transport, we can even talk about it tomorrow. There are amino acids and other things are also, they can be transported from the one side of the membrane, outside membrane to the inside. All the amino acids and sodium, all these things can easily come inside and active. Secondary active transport, it is due to the co-transport. In this case, a molecule that is moving naturally into the cell through diffusion is used to drag another molecule into the cell. And in this example, glucose hitches a ride with the sodium. That is, sodium is going along with the glucose. That is secondary. Now, as I have told you, endocytosis and exocytosis. Endocytosis means that is which is coming from the outside the cell, that is coming inside. The large molecules move in via one of the three forms, endocytosis or pinocytosis or phagocytosis. In this way, phagocytosis, you know how amoebal movement is and how they phagocyte material, dead material, and they bring inside. That is a macrophage system of the scavenging system of the cell. Receptor mediated endocytosis, as I have told you, these are all receptors. Where are the receptors? Just below the bilipid layer inside the sarcoplasmic reticulum. Extracellular fluid, it is coming down. Once it is coming in here, it comes in and vesicles are released inside, and again this break, again this unite. Here it breaks and again it unites. The pseudopodium movements, they engulf the material, they bring inside, and again outside membrane, it releases it inside, and outside membrane gets united again. Receptor mediated. Here are the receptors. Captured molecule, they are bringing inside and coated the bits and releasing it inside the vesicle. So receptors for mediated endocytosis. Neurotransmitters, enzymes, and for that the receptors are there. Cholesterol is taken up by this way. Exocytosis, that is from inside to outside. They are manufactured in the cell and released through the cell into the through the cell membrane, released outside through the cell membrane. So here, transport was like this transport, once it is coming nearer, it is releasing it outside and again uniting. From inside it is going out. Moved out of the cell cycle that fuse with the plasma membrane. How these are coming from inside the Golgi apparatus. From Golgi apparatus, they are coming outside and forming a vesicles. These vesicles, they are coming out. That's desmosomes. And these desmosomes, they are going out. Once this is out, again there is a fusion of the membrane, that is exocytosis. As we have seen, exocytosis and chemical combination, that is in the synapse, that is neuromuscular junction. In the neuromuscular junction, here it is a neuron. In this neuron, this is called as axon, that is a membrane. In between, there is a synaptic cleft. Here are the vesicles which are coming down and they are falling into the synaptic cleft. Here the acetylcholine receptors are there. These are the acetylcholine receptors. Initiation of the new impulse, they are dragging inside, and neurotransmitter substances, that is acetylcholine, is going inside the cell. There is a synaptic, there is a change in the synapse. That is neuromuscular junction, how the impulse is traveling from nerve to a cell. Vesicles containing neurotransmitter molecules such as acetylcholine. This is a synaptic vesicle. This is a neuron in between the cell and the axon. Here there is a synaptic cleft. These acetylcholine, once they are released, these are broken down, so they are getting attached to the neurotransmitter receptors. From there, they are taken inside. Vesicles, how they are coming and they are going out. How molecules cross the membrane? Diffusion, passive, osmosis, passive, facilitated diffusion, that is a passive way of diffusion. Now you have seen passive diffusion doesn't need energy, only protein needed. Direction, down gradient, temperature high, all these things are gradients which we have read. This is on in a tabular form. So, thank you for you all that you have listened the lecture very nice. So, what all we have read in the cell and cell membrane, they are same thing, how they are transported from outside to inside. That is called as transport across the cell membrane. What are the things we have taken in? It has to go inside. Thank you all.